Hello everyone. Today, let's discuss about circular dichroism spectroscopy. I'm Apoorva Shankar, Assistant Professor, Christy Jayanti College from the Department of Life Sciences. So what is circular dichroism? So circular dichroism spectroscopy, like our normal spectroscopy, is also based on Beer-Lambert's law. So here we are measuring the absorption difference between the two different types of light, that is the left circular polarized light and the right circularly polarized light. So this difference between the two is what is helping us detect what is present in our sample. So circular dichroism is an absorption spectroscopy method that is based on the differential absorption of the left and the right circularly polarized light. So we all know that there are two different types of lights, that is the linear polarized light and the circularly polarized light. So what is the difference between the two? The linear polarized light, the direction of the electric vector here remains constant, while that of the magnitude of the electric vector keeps varying. Whereas in circular polarized light, the direction of the electric vector keeps varying and the magnitude of the electric vector is constant. So this is the two main differences between a normal linear polarized light and the circular polarized light. So here we have two different types of circular polarized light. One is the right-hand circular polarized light and the other is the left-hand circular polarized light. So in the right-hand circular polarized light, we can see that the movement of the circular polarized light is clockwise, while in the left-hand circular polarized light, the movement is in the anti-clockwise direction. So this forms the basis of our circular dichroism spectroscopy. So here, let us understand the different types of instruments which are present in a circular dichroism. So here we have a light source. The light source can be a normal light source, normal white light source, which produces the wavelength in light in different wavelengths. And then we have a filter wheel, which serves as a monochromator to produce a light beam of a particular wavelength. And then the next part of the instrument is a polarizer, which is a very important part, which helps in converting a linear light into a polarized light. After which the polarized light passes through a quarter phase plate to produce two different types of circular polarized light, which is your left-hand circular polarized light and the right-hand circular polarized light. And then we have a sample cuvette, which helps in holding the sample, followed by the detector, which helps in detecting the difference in the absorption between the two types of light, which is the right circular polarized light and the left circular polarized light. So let us see the working here. So this diagram helps in the better understanding of the working. The S here is our light source, which produces light of different wavelength, and our monochromator, which is a prism, helps in producing single wavelength of light, followed by passing through the polarizer, which helps in producing right and left circular polarized light, which are indicated by the red arrows in the clockwise and the anti-clockwise direction. So this right and left circular polarized light passes through the sample cuvette containing the sample. So in case our sample contains more of left absorbing species, what happens is that the light that is transmitted will have a decreased in the wavelength of the left circular polarized light, which as you can see in the figure here. So here you can see the incident light. You have right and the left circular polarized light. which upon getting absorbed by the species which are present in the sample, you can see that the circular polarized light has decreased the wavelength, whereas the right circular polarized light remains constant. So this difference depends upon what kind of absorbing species is present in our sample. And this difference in the absorption between the right and the left circular polarized light is what forms the basis of circular dichroism spectroscopy. And this is detected by the detectors. So I hope this principle of working is clear. So we have a sample which produces the light of different wavelengths and the monochromator which produces a single beam of wavelength in the desired wavelength, which passes through the polarizer which produces the right circular polarized light and the left circular polarized light. These two pass through the sample contained in the cuvette, and depending upon what kind of absorbing species is present in a sample, there is a difference in the absorption in the transmitted light, and which is detected by the detector. So this circular dichroism is also depends upon the principle of Beer-Lambert's law, like our uh, any other spectroscopy methods. So a Beer-Lambert's law state that there is a linear relationship between the concentration and the absorption of the solution. So here what happens is that the difference between the absorption of the incident light and the absorption of the transmitted light is what forms the basis here, even in the circular dichroism spectroscopy. So the applications. So this is one of the output of the circular dichroism spectroscopy, showing the different types of secondary structures of protein. That is the alpha helix, beta helix is what we can see in the graph here. So this is based on the differential absorption of the right and the left circular polarized light. So what are the applications? You can study the different secondary structures of protein using this type of microscope and also this type of spectroscopy, sorry. And you can also study the influence of various external factors like pH, solvent, and temperature on the structure of the protein. So the structural changes which are induced by the various factors can be studied using circular dichroism spectroscopy. The various patterns of folding and unfolding can also be determined, and the influence of ligand and ion on the structural changes can be determined, and also the structural features of chiral compounds can be studied using circular dichroism. So I hope this concept of circular dichroism is made simple and understandable. Thank you.